Okay. Can you see my screen, guys?
Yes, ma'am.
Yeah, yeah.
Okay. So any doubt in the pipeline, like this in the steps? Is it clear? So there are some important terms that you should know before running this reference based pipeline. So what is this biological analysis plan? Why we need this? Because in order to check the gene expression, we need what? The biological analysis plan. So yesterday also I told you what are those two plans, two universal plan. Tell me. Any one of you?
Control versus treated and treated versus control, ma'am.
Yes, ma'am. Right. So control versus treated, treated versus control. So if you have this control versus treated, that means you are going to check the expression of control with respect to treated. But when you have this plan, then you are going to check the expression of treated versus control. So let's understand how to check the gene expression. So for example, I'm having a gene 1, gene 2, gene 3. This is my control and treated. So these are my read counts of my control. I'm just taking an example. Now, I've taken two plans, for example, control versus treated. And treated versus control. Now, if I want to check the expression of control versus treated, so what I will do, just write here equal to and then control divided by treated.
We don't see the
Excuse me,
screen, your
ma'am.
screen.
Yes, ma'am. Ma'am, yeah, the screen is
You
not
can see.
visible. We are just
No.
seeing the Excel workbook.
Uh, just wait. Now, sir, can you?
Yeah,
Just
now we
refreshing.
can. Yeah, now
Yeah.
we
Can you see now?
Yeah. Yes. Yeah.
Anirudh, sir, Akansha, ma'am, can you see my screen?
Yes, thank you.
Okay. So I told you on this A column, these are my genes. I'm taking just an example of three genes here, gene 1, gene 2, gene 3. So this is my control sample and these count are my reads. Okay. And for this treated, again, this is for the treated sample. These are my read count. Now, there are two plans, control versus treated and treated versus control. So if our plan is control versus treated, so how to check the gene expression? You just have to type equal to, then control divided by treated. Then enter. Then what you can do, autofill your data. If your plan is treated versus control, so here you have to type equal to treated divided by control. Then enter. Again, you can autofill this. So for this, you are having the gene expression. So can you tell me which gene is highly upregulated and the downregulated? Can you tell me? Can you tell me, guys? By looking at this data.
Gene 3 is upregulated.
In 3,
Yes,
this
ma'am.
one.
Yes, ma'am.
Okay.
And gene 2 is downregulated.
Gene 2. Okay. So see, like in this Excel, you are just having the 3 genes, right? It seemed like some of the genes are upregulated and the downregulated. But what if you are having 1,000 of genes or maybe more than that? It's very tedious task to do it manually, right? And you cannot tell me like by looking at this data, this is my upregulated and downregulated. So what they have applied, so one method they have applied, that is the log2 fold change. So I will tell you why. So let me just write, this is for the control versus treated. Have you ever heard about this log2 fold change? What is that?
No, I didn't.
No, ma'am.
Okay, no problem. So what is this log2 fold change? It is going to quantifying the difference between the sample. And here, by calculating this log2 fold change, we are going to get the result in the form of positive and negative values. Okay. So this positive value, it is going to indicate what? Any idea?
Upregulation.
Yes, upregulation. Whereas this negative will indicate my downregulated genes. So this fold changes what? Whenever you are going to compare one expression with respect to another, that is the what? The fold change. Okay. And it is going to quantify the difference between the sample, and by just calculating this log2 fold change value, it is very easy to predict what is my upregulated genes and the downregulated genes. Okay, so let's see how to calculate that log2 fold change value. So there is a formula. You have to put here equal to log. So our plan is what? Control versus treated. So you already calculated this control versus treated. So you have to take the value from here, then comma 2. Then put the brackets, enter. Then for this, again you have to put here log, put the brackets. Now which value should be taken? This one, treated versus
T2.
control. Then comma 2, bracket close, enter. Now, can you see the minus sign here? So this minus sign is clearly indicating these are my down regulated genes, whereas the positive values will indicate my up regulated genes. But make sure you can see whenever you are having the plans control versus treated, the treated versus control, the results are different. Can you see that? So
Yeah.
just make sure whenever you are dealing with your plans, you have to be very carefully, like either you are working on treated versus control or control versus treated. It will be different. Okay. Now, for this log to fold change value, there are some standard cutoff. If your log to fold change value is greater than or equal to 1, those are my upregulated genes. And if the log to fold change is less than equal to -1, that are downregulated genes. And if your log to fold change value is equal to 0, that means there is no gene expression. Are you getting this?
Yeah.
Yeah, thank you.
Okay. So now, can you tell me for this gene 1, which is my upregulated genes?
The G3,
G1, sir.
G1,
Yeah.
It's downregulated.
Down-regulated.
No, it's upregulated.
Which one, sir?
it depends.
The
Like,
one.
the
If we
treatment
come,
versus control, maybe it's upregulated or
Yeah. So you can see here, gene 1 is showing the higher expression here in the treated versus control plan, right? In this. And you can
Yeah.
also check the expression from here also. Treated versus control is 2 here. Can you see? And also, if you want to cross check your data, why it is upregulated in the treated versus control, you can see the treated reads here. There are 20 reads. Okay. So in this biological like analysis plan, this log to fold change values is very important. So did you got this? What are the standard cutoff? Like, because in the data analysis we are going to see this. Okay, is it clear why we are calculating this?
These cutoff value we only apply for the treated versus control, na?
No, ma'am. So it depend on your data. These are some standard cutoff, ma'am, for the any plan. Maybe on your like any plans you can use it for this. Okay, but these are standard cutoff, but it depending on your data we can also like shift the cutoff also. Okay.
Means when we take the control versus treated, it G1 have -1,
Mm-hmm.
then it is down-regulated.
Yes, ma'am. I told you, for example, like multiple genes, okay, maximum genes, they are having what 0.9 value. That time cutoff value you cannot put as the greater than 1. Why? Because it is going to show you 0 values, right? For the genes, for the upregulated genes. So if 1000 of genes is having the log to fold change as a 0.9, that time the cutoff will be what? The 0.9 there. Okay? Clear?
Okay.
Depending on your data, ma'am.
Okay.
Yeah. Did you got this idea regarding the upregulated and the downregulated?
yes.
Okay. Now, what are the replicates? Can you tell me?
The
C1 and
duplicate
C2.
copies.
Yes. So it is a you can see, you can tell, like, it will check the variability in the sample. Or you can say there is a repetition of experiment on a particular sample again and again. So that is what the replicates. So there are two type of replicates, that is technical and next is the biological replicate. Do you have any idea about this technical replicate and the biological?
Yeah.
Can you tell me, sir?
Yeah, the technical replicate, it's like, I will take an example to explain. If you can have an experiment today, let's say you want to treat cell to, let's say with a drugs to see if maybe it help to cure a disease. You can make three plate. If it is cell line, you can make plate 1, plate 2, plate 3, and you consider for each, let's say if you have control where you put nothing and the treated condition where you put something, you have that in 3 copy, so that each condition will have 3 we have 3 each condition will have 3 sample run at the same time in the same
Yeah,
condition.
yeah,
Yes,
yeah.
that's what we call
Yes,
technical replicate.
yes, sir.
And now the biological is what we call independent replicate. When you can run one experiment today with its technical replicate, tomorrow you run another one, the same experiment in the same condition, but different day.
Yes, sir. Right. So let's discuss about this technical replicate. So for example, you are having a leaf here and there are like multiple cells in this leaf. So what I will do, I'm going to isolate this particular this cell. Okay. This one. So this cell is isolated. Then what we are going to do after isolation, different experiment will be performed on this particular one cell. Okay. So that is what the replicates. We are performing the repetition of experiment on this particular one cell and you are getting what? The technical replicates here. Okay. Did you got this? So technical replicates means same experiment we are going to perform on sample again and again. Okay. That is what the technical replicate. So mainly it is going to focus of your research is like when you are assessing one sample, then you are running the technical, that is what technical replicates. Whereas this biological replicate, as sir said, you are having the, for example, so you are having two hibiscus plant here, and from this plant I have isolated a leaf, and then again on this leaf, like you are having the cells, different cells. Then this A cell and this B cell I'm going to isolate, and again I'm going to perform the experiment on this particular cell, and you will get what the replicates. So this is what the biological replicate. So we are taking the different biological sample within the same population. That is what the biological replicate. Did you got this difference between the technical and the biological?
Yeah.
No, ma'am. Please, can you repeat?
Ma'am, like in the biological, you are having the two different plant. Okay, can you see the two hibiscus plant here? Yeah.
Okay.
From this A plant, I have taken the cell. Here you can see it has been taken from tree 1, and this is my tree 2 here. Okay. But whereas in the biological, or you can see the technical here, what I show you, there was a one leaf. Okay. So in the leaf, again, like there are different cells. So I have isolated a cell from this leaf, and then I'm going to perform the experiment on this particular one cell, okay, multiple times. And
Okay.
then you are getting what? The replicates, right? So
Okay.
these are what? The technical, ma'am. But in the biological, we are taking two different biological sample within the same population. Okay? That is biological.
Okay.
Okay?
Okay. Okay.
Now, there are important terms in the NGS. So first is the type of sequencing. Can you tell me what type of sequencing we are doing?
You mean the long read and short read, or
Okay. Single end
single and parent? Yeah.
sequencing, and next is paired end sequencing. So first, let's discuss what is the single end sequencing. For example, this is a strand here. Okay. Then there are forward strand and there are reverse strand, right? So
Yeah.
in the single end, what happen? We are ligating a adapters. Okay. So adapters are ligated at one end. Okay. Then it start doing what? The sequencing machine, it is reading. Okay. Then for this particular forward strand, you are getting what? The forward file here. And this is my reverse strand. Can you see?
Yeah.
So if the adapters are ligated at the reverse end, okay, again the sequencing machine, it will read and it will give you the reverse file. So either you will get forward or the reverse file, okay, depending on this data. But whereas in the paired end, what happened? Adapters are ligated at both the ends here. And sequencing machine, it will start reading from this side also and from here also. And in the paired end, you will get two files, okay, forward and reverse. These two files you will get, and this forward file will be indicated by _1, and this reverse file you will get by this _2. So in the paired end, remember, you will get these two files. So can you tell me which type of sequencing is better, single end or the paired end?
Parent.
Why, sir?
Maybe because it covers all fragments.
Yes, sir. Like in the paired end, why it is better? Because you can see it is reading from both of the side, right? So we cannot miss any of the information in the paired end data. So that is why paired end type of sequencing is better than the single end. Got it?
Yeah.
Next is depth. Any idea about this depth word? Have you ever heard this depth word?
Number of times the sequence
Yeah.
it is sequenced.
So how many times the particular base is read by the machine or sequenced by the machine? That is what the depth. For example, this is my reference genome. And I'm going to align my reads on this reference genome. So it will align like this. So what I saw on this, maybe on this 20th position on this reference genome, I'm having what? The adenine. That is capital A on this reference genome. Okay. So, and what I saw on this entire reads on this 20th position, they are having what? The adenine. Right? So how many times the adenine was called here? Can you tell me? Any idea?
6 times.
How 6 times, Rakhi? Like, see, there are total
10
10.
times.
Yeah, right, sir. Who is this?
Anirudh.
Yeah. So you can see there are total 10 reads here. And this one read, it is calling A here. Then 3, 4, 5, 6, 7, 8, 9, 10. So there are 10 times this adenine was read, right? So for this particular read, what is my depth here? 10x. Why 10x? Because adenine was read 10 times by the sequencing machine. Okay? Are you getting this depth?
Yes. You mean
Yes.
the coverage?
Not the coverage, sir. Not the
Mm,
coverage. Coverage is different, sir, from
okay.
the depth. Depth means how many times the base was read by the machine. So you can see here, if on this position, like maybe on this reference genome, you are having here guanine. Okay. And here you are having what? The
4.
t, maybe
Mhm.
t here. Okay. So from this data, you can also tell me this type of data, it is having the mutation. Why? Because on this reference genome, there was the G present here and it was getting replaced with the T, right? So here we are saying that it is calling the mutation here. Okay. Now, but what is the depth for this T here? Can you tell me?
Oh,
Yes, sir. That is 4. So you can see 1, 2, 3 and 4. 4 times the T was read by the machine. So here for the T, depth would be the 10x. Sorry, 4x. Got it? The depth word?
Yes, ma'am.
Okay.
And when one depth is 10x and other is 4x, what is the difference between them? I mean, is it relation to the our quality of our
Yes,
sequence?
sir.
Okay,
Yes, sir.
okay,
See, for example, like you are having the 4x, okay, or maybe the 10x. Here I was just explaining you like what is the depth, but there are certain cutoff for your depth also. So minimum depth should be the 50x, sir, for the RNA sake. So I will write here. So minimum depth for DNA sake is 10x and minimum depth for RNA sake is 50x, sir. Okay.
okay.
Actually, this will give you the better confidence. Okay. So if, for example, you are having the reference based data and you are having the 10x, 4x depth, that time you have to neglect those reads, sir. Okay. Because our cutoff is more than 50x, sir. Okay. Minimum depth should be for this 50x, sir. Are you getting this?
Yeah, just I have a curiosity when, for example, our our depth is 4, because
Mm-hmm.
it's comparing to the our reference genome. Yeah,
Yeah.
if because you told now we should forget about that sequence that is for amount. I mean the quality. If for example we change our reference genome, it cannot be okay then.
No, sir. Why to change
Mm,
the reference, sir?
maybe just a curiosity. Maybe if
No,
we
no, no,
change.
no, 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 no,
Mhm,
sir. Actually,
mhm.
for example, your data is for the human,
Yeah,
right?
yeah, yeah.
And you are taking the reference genome as for the Drosophila. So it is not going to map the reads over
Okay,
there,
okay,
right?
okay, okay.
So it should
Understood.
have same reference genome. Then only it is going to map, right?
Okay,
Yeah.
okay.
Can you please re-explain the depth from where you were talking about cutoff? I did not get it properly,
No, no, no,
please.
sir. Actually, the depth mean here is like how many time the base was read. So what I'm is going to explain you that what is 10x here. You can see on this position adenine was read 10 times, right? So here for this particular read, this is what 10x depth, right?
Yeah.
But here you can see on this position the t was called 4 times, that is why
Yes.
I have written 4x depth.
Yeah.
Okay, clear?
Yes.
Clear about the depth here?
Yes.
So we will see like in detail, like further in the pipeline, when this depth, coverage, mapping quality will come, you will get the idea, because there you will get the figure over there in the result. Okay. But
No,
for now, yeah,
I mean the
then
cutoff.
cut off this one, 50x. So I'm telling you, whenever like you are having the reads with the 10x depth, you are having the lower confidence, sir. Okay.
Okay.
Yeah, so we have to neglect, we are putting some standard like cut off theirs. Okay, we cannot take the low reads which is having the low sequence also, we cannot, we have to neglect those. Okay, otherwise
Okay.
it will create a noise in your data also. Okay.
Yeah.
Yeah, we will see in the pipeline how to neglect those things. But the idea is like, the is it clear what is the depth word
Perfect.
here?
Perfect. Yeah.
Okay, fine. Now coverage means how many reads are able to cover the reference genome. That is what the coverage. So for example, this is my again reference genome, and these are some reads which is covering the reference genome. And for this data, my coverage is 50%. Okay. You can see the 50% of the reference genome, it is not covered here. Can you see?
Yeah.
Right. So coverage means how many reads are able to cover the reference genome. So you can see the 50% of the reads are covering my reference genome here. But if I increase the depth, so what it will do, it will increase the reads here, and it is going to cover your reference genome. So that means by increasing the depth, it will increase your what? The coverage.
Coverage.
Okay. So that means coverage is directly proportional to depth. If depth increase, coverage is also increasing. Clear about this depth and the coverage?
Professor, this increasing depth is the method that we do, or we ask from the company that we are sequencing?
Yes, sir. We have to ask for the company, sir. We
And
whenever
mhm,
we are like sending our data, we have to tell to the company only what
yeah,
are the depth parameter or the cutoff for your mapping quality you are going to put. You have to tell those to the sequencing people.
yeah, and if we, for example, ask them to increase the depths, it means they will charge us more. Yeah.
Yes, sir. Yes,
Okay.
sir.
Okay. Thank you.
Excuse me, ma'am.
Yes, ma'am.
Ma'am, in coverage, we covers the read.
Yeah.
How many reads are able to cover it? So reads are the replicates of the reference, and in depth we saw there are the lot of reads who have same base pair that have our reference. So coverage is what means how many reads cover it. But in
Yeah.
depth we have a particular base that say different reads have. So how
Yes.
could we say that coverage is directly proportional to depth?
No, 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 no. See, if you are going to increase your depth, that means more number of reads will be covered there, right? For example, you are having the 4x there. That means the 4 reads they are covering at particular one position, right? That is why we are taking it there as a 4x, right? If I choose 50x, that means 50 reads are covered there, okay, in the coverage parameter. But in the depth, on the particular one position they will take, ma'am. Okay, but the 50 reads will be covered in the coverage, ma'am. Are you getting this? For example, depth is telling you about how many times the base is read on this particular position, right?
Okay.
So here you can see it is reading 9 times the base was read here.
Okay.
Yeah. So this is what 9 times the base was read, right? But along with that, they are taking this reads, entire reads also, ma'am. Okay.
Okay, that covers that particular
Yes, ma'am.
area.
Yes, correct. That it come under the coverage, ma'am.
Okay.
Okay.
Okay. Thank you so much.
So what is this Fred quality score first? It is assigned to each and every base which is sequenced by the machine. So Fred quality score is what? It is assigned to each and every base. That means in the data, each and every base is sequenced by the machine and some quality score is given to your base. So cutoff, I will tell you later on. So let's discuss what is this Fred quality. Yeah, can you see this table?
Yeah.
You can see on this first column there is Fred quality score, then the probability of the incorrect base call and the base call accuracy. So for example, we are having the Fred quality score as the 10 here. So 10 quality score is indicating what? The 10 time the base was called and out of 10 time one base was incorrect. Okay. So that means 9 time the guanine was read and one time it showed adenine. So that is what 1 in 10 here. Then for that, the base call accuracy here is 90%. But if you increase the Fred quality score to 20, then what happened? the base was read 100 times, and out of 100 times, 99 time the base was read correctly, and one time it was showing what incorrect base there, and there
Yeah.
you are getting 99% base call accuracy. But if you increase to 30,000 time the base was called by the machine, out of that one time the base was incorrect, then base call accuracy was 99.9. So we are putting the Fred quality score cutoff as equal to 30 or more than 30. Why more than 30? Because if you increase Fred quality score before 30, you can see if it is moving to 40, 50, the base call accuracy is 99.99. Can you see? So none of them it is giving you what? The 100% base call accuracy, right?
Yeah.
So that is why we are taking the Fred quality score cutoff as the equal to, I will write greater than equal to 30. Are you getting this? 30 cutoff, why we have taken? Because again you can see here, after 30 the base call accuracy 99.9 only. Okay, it is not reaching to 100%, right? So that is why we are taking this as a standard cutoff for the Fred quality score. Did you got this Fred quality score?
Yes.
So what is this BAM and SAM file format? So these are two type of file you will get during the,
Amen.
yeah. So it will, it is containing what? The mapping information, or as you he said, the alignment information. So this BAM stand for binary alignment map, which is what? Computer readable file. Whereas this SAM file stand for sequence alignment map. This is your human readable file. So whenever you want to read the file, if I want to know what are the content that is present in my file, so that time you should have the SAM file, right? But this SAM file, it is not readable by the tools or maybe the computer. Okay. So that time you have to convert this SAM into the BAM file. Why? Because this, it will contain what? The binary file that will be readable by the computer. Okay. So that conversion you can do with the help of one tool, that is SAM tools. So SAM tools, what they do, it is going to convert your SAM to BAM file or BAM to SAM file. Okay, got this? BAM and SAM file.
Yeah.
Next is cigar string value. Any idea regarding this cigar string value?
No, ma'am.
Okay. So it will tell you how bases from the read is aligned to your reference genome. So let me tell you with the help of diagram. So this is the read. It is aligned on this reference genome and it is having 50 base pair. So cigar string will tell you how the base from the read is aligned to your reference genome. So for example, this 50 bases is aligning, or you can say it is matching with your reference genome. It will give you what? The cigar string value as the 50M. So this M is what? The associated operator. So here M is indicating what? The 50 bases of from the read, it is matching with your reference genome. But if only 40 bases are matching, then it will give you 40M. And out of 10, the bases are deleted. So it will tell you 40M here and 10D. So D stand for deletion. Okay. So 50M is telling you 50 bases are matching. Here, if you are getting this cigar string value as this 40M 10D, that means 40 bases are matching and the 10 bases are deleted. Next, if you see like this cigar string value, so 40 again it is telling you 40 base are matching, 5 are deleted and 5 are inserted. Clear about the cigar string value?
Yes, ma'am.
Yes, ma'am.
So mapping quality is what? How read is aligned to your reference genome. That is what the mapping quality. So this is your reference genome. Okay. So there are some reads which is matching on this reference genome. And so can you tell me out of this three read, which read it is having the good mapping quality?
The middle one.
Yes, sir. The middle one. Why? Because the 80% of the read, or you can see the more number of bases are there that are covered or that are matched on this reference genome from this, right? So that time you have to take this as the mapping quality, 80%. Clear? The mapping quality.
Yes,
So
ma'am.
it will just give you just a percentage of the read, how read is like aligning on your reference genome. Okay, that's it. Clear about this terms?
Yeah,
Any
it's
doubt?
And Professor, what about that two other person. What should we do with two other person? I mean the 20% and 60%.
Which, sir?
At the paint yet that you put.
Mm-hmm. Yeah, this, sir.
Yeah, yeah. I mean,
Again, like in the mapping quality, we are taking cut off
mhm.
equal to or more than equal to 30, sir. Here also. Okay.
Okay.
We want the good quality of our data. So make sure, like for the Fred quality score and the mapping quality score, it should be always greater than equal to 30, sir. Okay. Below that, we are just discarding all those reads, sir.
Okay. And just sorry, how we realize that this cutoff has more than 30%? Is it any formula or?
Yeah. No. Like we are putting the command there in the tool,
Okay.
sir. Whenever
Okay.
like we are
Okay.
running the tool, we are just, I will
Okay.
tell you, I will guide you,
Okay.
okay,
Thank
during
you.
the
Thank you.
installation. Yeah.
Okay. Thank you.
Yeah. Any doubt till here? Okay. So there are some tools that we are going to install for this reference based. So there are two methods. First method is the repository. Second is the source code. So first we are going to see the repository method. So what is repository? Any idea?
No, ma'am.
Sakshi, Rakhi?
I think that in the repository we can go in let's say we can go online in the same library in the same let's how do we call that, like in GitHub.
Yeah. So that
Yes.
GitHub is what the source code methods are. Okay.
Okay.
Yeah. So this repository means the storage location. That means tool which is already there in your Ubuntu storage. Okay. In the Ubuntu repository, that is we are going to install. So there are two commands: sudo aptitude search tool name. So what is a sudo here? So it is giving the permission to access root of your system. So it will install your tool like globally in your system. Okay, so you can operate your tool from anywhere. That is what the sudo here. Then aptitude is your package manager. That means it is storing all the information for all the tools. Search is the keyword search. Then give your tool name. Then how to install that tool? Sudo apt-get is to retrieve the package from the aptitude. Then after retrieving from the aptitude, it will install the tool. So
Oh, it's not yet installed.
first command is sudo aptitude search your tool name. So first tool name was what after raw data? Tell me. Fast Q, right? Fast QC tool. So you have to type here sudo aptitude search and give your tool name, then enter. Give your password. Then you can see in the Ubuntu storage, the FastQC tool is present. Can you see FastQC? What it is doing? Quality check, right? For your data. Then now we know in the aptitude, the FastQC tool is there. So we have to install this tool by running this command sudo apt-get install FastQC. Enter. It is installed. Then how to check whether it is installed or not? You have to type FastQC -help. So it will open the manual. So in the manual, it will tell you how to run this FastQC tool. So you can see the command here, FastQC, then give your file name. So this is how we are going to see the tool installation by repository. Did you got this repository method? Any doubt?
So will we go in detail about this install chain?
Yes, sir. Yes, sir.
Okay,
Yes.
thanks. Okay.
So I've just given you the brief description over here, like, but from tomorrow onwards, the pipeline will be started and I will tell you how to install the tools with the help of the command. And then also along with the tools, the result will be generated and all the results we are going to analyze, sir. Okay.
Thank you.
Yeah.
And ma'am, the
Hello.
data that you shared today in the
Yes.
morning,
Yeah.
it is still not visible. So will it be available on
So
the drive or what?
yes, sir. I have already shared in the drive also. You can have that access from there. Okay.
Let me see.
Yeah. So again, like whoever like the WSL, it is not there. Just ping me personally. I will connect you tomorrow, maybe before the session. And whenever you guys are free, just let me know. Okay. Then we'll connect. Okay, then we'll meet you tomorrow, guys, at same
Thank
time.
Thank you.
Okay. Okay. Bye bye. Yeah.
Bye.
Excuse me.
Yes, sir. Yes, sir.
Yeah, you said at what time tomorrow can we connect if the
Uh, okay. So
WSL has not yet?
whenever you are free, just let me know. Just ping me on my WhatsApp group.
Okay, perfect.
Yeah, maybe on my personal contact number, you can just text me.
Yeah, I'll ping you even maybe today if you don't sleep yet.
Okay, for sure. Yeah.
Perfect. Thank you.
Okay, bye bye.